This is a distance learning modification for medical interventions, lesson 1.2.3, attack of the superbugs. So in this lesson, what we are attempting to do is to take two different strains of E. coli that's each resistant to a different antibiotic and allow them to undergo conjugation to produce a new strain of E. coli that is resistant to both of the antibiotics that we are interested in. So antibiotic resistance is caused by genes for specific processes, and it could be efflux, a little pump that pumps antibiotics out of the bacteria cell before they can do harm. It could be inactivation destruction, where the bacteria has a gene for making enzymes that can break down antibiotics before they can do damage. Or it could be an alteration to one of the surface antigens that antibiotics have to be able to attach to in order to enter the bacteria cell. So bacteria cells can share these genes with each other, and that's how we can have a non-resistant strain of bacteria become resistant. It accepts a gene from another cell that is able to pass that gene on. Okay, so genes that are on chromosomal DNA, they can be passed on via processes like transduction or transformation, but not conjugation. This DNA is much too large to be passed along by conjugation. You'll see here in a couple of minutes why. Plasmids, on the other hand, are very small circular pieces of DNA that can pass by conjugation. And so that is what we are going to be experimenting with in this activity. So in conjugation, you have your two different bacteria, and one of the bacteria, who's the donor, has a plasmid that it wants to pass along to the recipient cell. So it's going to extend a pilus to attach to the recipient, and then they'll get reeled together by the pilus so that they're in contact with each other. And they kind of form a little tube, a little conjugation tube that is going to allow the passage of the plasmid from one cell to the other. So you can see that this plasmid, it's going to unzip, and then it's going to allow half of the unzipped plasmid to go to the recipient cell. Once that occurs, both cells will replace the other half of the plasmid, and then they'll detach from one another. So you can see that this plasmid now is a part of both of the cells. So we're going to be doing an experiment on paper and kind of be going through the steps that you would be doing in class to show conjugation happening between two strains of E. coli so that we make a new strain that has a resistance to two different antibiotics. So here are our strains, and this is information that you need. We have what we're going to call E. coli 1. It's resistant to an antibiotic called streptomycin, and the gene for its resistance is located on the chromosomal DNA. The second strain is E. coli 2. It has resistance to another antibiotic called ampicillin, and this gene is located on the plasmid. And so we're going to ask you in this lab to think about what I just told you about conjugation and make some predictions, and then we're going to carry out the experiments. So there is a copy of a slide presentation on Canvas that you will be putting your information into. So as we go through this presentation on this video, you're going to transfer this information over to that slide, to slides on that slideshow that you've got copies for on Google. Okay, so the purpose of this experiment is to observe how bacteria can share genes for antibiotic resistance via conjugation. We're going to be dividing this experiment up into 3 days. In day 1, we would be growing the bacteria on plates of agar that contain the antibiotics that they're supposed to be resistant to, and then allowing them to incubate and confirm that the E. coli strains showed the type of resistance that we were told they would. So we'll call those the confirmation plates. On day 2, you're going to take some of the E. coli 1 strain and E. coli 2 strain, and you're going to mix them together on one plate. And hopefully they'll undergo conjugation. So that would be day 2. We would allow them to incubate overnight. And then on day 3, we're going to again get plates that contain antibiotics and grow the new strain that should have occurred from our conjugation plate on those different plates with antibiotics to determine whether or not conjugation was successful. Okay, so here is what you need to do for day 1. On day 1, again, the purpose of our experiment on day 1 is to confirm that the bacteria do what we were told that they would do. And that is that E. coli 1 is streptomycin resistant and E. coli 2 is ampicillin resistant. So we're going to carry out the day 1 portion of our experiment. You're going to need some paper and some colored pencils. So if you would, you might want to pause and go ahead and do that. And we're going to start out by drawing 4 circles to represent nutrient agar plates. Oops, color pencils out. Okay, so this is going to be day 1. And again, these are confirmation plates. And again, what we're confirming is that the bacteria are resistant to the antibiotics in question. Okay, so you have 4 plates. 1, 2, 3, 4. Okay, the first plate just contains regular agar, and we've added some special nutrients to this agar that is good for E. coli bacteria in particular. And it's called luria broth. So we're just going to put LB for luria broth agar. All right, so this first plate just has yummy stuff that the bacteria like to eat. The second plate, it's going to have luria broth agar, but we will also add streptomycin, which again is an antibiotic. So it's got LB agar plus it's got streptomycin. Okay, plate 3, it's going to have LB agar in it. And it's going to have ampicillin. Ampicillin is another antibiotic. It's actually a beta-lactam like penicillin. Okay, and in our 4th plate, we're going to have the LB agar and both antibiotics. And I'll just put streptomycin. I'm going to put strep for short and amp for short. Ampicillin. Okay, y'all. At this point, I want you to stop and I want you to make predictions as to what should happen in these plates. Okay? Well, before you do that, sorry, let's go ahead and mark up these plates. We're going to divide each plate in half. And on one side, we're going to streak a little bit of E. coli 1 bacteria. And on the second side, we're going to streak a little bit of E. coli 2. Now remember, E. coli 1 is supposed to be streptomycin resistant, and that's located on the chromosomal DNA. And remember that E. coli 2 is supposed to be ampicillin resistant. And that gene for ampicillin resistance is supposed to be on the plasmid in this bacteria cell. Okay, we're going to do the same thing for all four of these plates. We're going to streak a little bit of E. coli 1, a little bit of E. coli 2 on both plates, or both sides of the plates. Okay, now, sorry, at this point, I want you to stop and make predictions. So LB agar, there's no antibiotic in this plate. We're going to streak a little bit of E. coli 1 here, E. coli 2 here. Will the E. coli grow on both sides of the plate? Yes or no? Make a prediction. Okay, same thing for the rest of the plates. Okay, E. coli 1 is streaked over here, E. coli 2 is streaked over here, but this plate contains some streptomycin. Will the E. coli 1 grow? Will the E. coli 2 grow? Make your prediction. All right, and then go ahead and do the same thing for the other two plates. All right, and keep in mind what they contain, and then keep in mind what they're supposed to be resistant to as you make those. All right, so pause the video and write down your predictions somewhere. Okay, I'm going to assume that you have paused the video to make your predictions and now you're ready to move along. Okay, so what would actually happen in real life? Well, if we were in class, if everything worked according to plan, on the LB agar, we would have bacteria grow on both sides. So I'm just going to use purple to represent bacteria. So we would have bacteria growing here and we'd have bacteria growing here. Okay, on plate 2, remember this plate contains streptomycin mixed in with the agar. E. coli 1 is resistant to streptomycin, but E. coli 2 is not. What that means is that E. coli 1, since it's resistant, it doesn't care. Streptomycin could be there, big deal. It's not going to bother it. So E. coli 1 will grow because it's resistant to streptomycin. On the other hand, E. coli 2, no growth. Streptomycin kills it or keeps it from growing. All right, plate 3, we could remember this one contains ampicillin, so we would see that the bacteria would not grow over here because E. coli 1 is strep resistant, but it's not ampicillin resistant. But this guy, yeah, he likes it. He's okay. So we should have growth over here, but no growth over here. Okay, I'll even write no growth. No growth to emphasize that. And then over here, guys, well, we've got both streptomycin and ampicillin mixed in with this plate. So this guy can't grow because the ampicillin that's in there, and this guy can't grow because of the streptomycin that's in there. So it would be no growth on both sides. Okay, so that's day 1. You're going to use this information. You need this picture, and you're going to take a picture of this and you're going to paste it into the appropriate slide on the Google slideshow. You also need to fill out the chart that has your predictions on it and the actual results on it. All right, so now we're ready for day 2. So we're going to make a new drawing, but for day 2, we're going to take this plate right here that has both the E. coli 1 and the E. coli 2 growing on it. And we're going to mix them together. So this is going to be day 2. And the purpose of day 2 is we're going to allow these two guys, E. coli 1 and E. coli 2, to mix together on the same plate. We're going to mix them all together and allow them to undergo conjugation. All right, so day 2, we're going to have a single plate, and we're going to take our plate 1 from the previous day. And we'll put that as our day 1. And remember, we had growth on both sides. We're going to take some of E. coli 1 and put it on this new plate. And this is our conjugation plate. And all it contains is LB agar. There's no antibiotic in this one. Okay, so we've taken some of our E. coli 1 and put it on here. And now we're going to take some of our E. coli 2 and put it on there and mix them all together. Okay, and then we're going to let them incubate overnight. And hopefully what's going to happen is they undergo conjugation. Now remember, E. coli 1, it's streptomycin resistant. That gene is located on the chromosomal DNA. E. coli 2 is ampicillin resistant. That resistance gene is located on the plasmid. So supposedly what will happen is that the plasmid from E. coli 1, or sorry, E. coli 2 will be shared with E. coli 1. So the purpose of this day, the purpose of this plate is to allow conjugation to occur. And we call this our conjugation plate. All right, you're going to take a picture of this and you're going to paste your picture into the appropriate slide on the Google slideshow. All right, so we're ready for the final day, day 3. So we are going to need our day 2 conjugation plates. We need this plate. And we're going to set up more agar plates. This time, they're going to have the antibiotics in them. So this is going to be day 3. And we're going to confirm that conjugation occurred and that we have a new strain of bacteria that hopefully is resistant to both types of antibiotics. Okay, so confirmation of conjugation. All right, so we're going to need our conjugation plate. And remember, it just had LB agar on it, so we should have a nice bunch of growth here where we mix together E. coli 1 and E. coli 2. And we're going to need 3 more plates. Okay, and plate 1 is going to have our LB agar, and it's going to have streptomycin mixed in with it. Plate 2 is going to have the LB agar and ampicillin. Both of the ampicillin is going to be mixed in with the agar on this plate. That's plate 2. Plate 3, it's going to have both antibiotics mixed in, kind of like we had on day 1. And I'll just put abbreviations, strep for streptomycin and amp for ampicillin. Okay, so what we would do on this day is we're going to take some of this bacteria from the conjugation plate and we're going to put some on each of the different plates. And we're going to incubate them and check them for growth the next day. All right, so at this moment, I want you to pause the video and I want you to jot down your predictions. Will this bacteria from the conjugation plate, will it grow on a plate that contains streptomycin? Yes or no? Will this bacteria grow on a plate that contains ampicillin? Yes or no? And will it grow on a plate that contains both? All right, so take a pause, think about it, and write your predictions down. Okay, we're back. So here's what would happen. Well, if conjugation was successful, then yes, we should see growth on the plate that contains streptomycin. And yes, we should see growth on the plate that contains ampicillin. And guess what? We should see growth on the plate that contains both. And that's because when we underwent conjugation, we shared the plasmid from E. coli 2 with E. coli 1. E. coli 1 already had the gene for streptomycin resistance on its chromosomal DNA, and now it also has the plasmid that has the gene for resistance to ampicillin. So it's got both. So we have a new strain of bacteria that's resistant to 2 different antibiotics from plate 3. Okay, so you're going to take a picture of your day 3 drawings and paste those into the Google slideshow, and you're also going to complete the charts. The Google slideshow also asks you to provide a problem statement, some background information. Basically, anywhere there's green text, you need to type responses. Those are prompts for you to reply to. All right, if you have any questions, of course, you can email me and let me know and ask for some clarification. I hope you have a great afternoon.